Hi, this is Francesca Peri, talking from the MBL in Heidelberg. In the lab we are interested in microglia and their many functions in the brain. Microglia are the macrophages that populate the vertebrate brain, and what these cells do is quite remarkable. They constantly scan the central nervous system looking for apoptotic, sick, but also injured neurons, and they remove these cells with great precision. And for example, in the case of neuronal injuries, this is extremely important to allow the subsequent brain regeneration process. Of course, it's of great importance to understand how microglia can identify these targets within the large brain, and how the underlying signaling molecules travel, propagate across the brain to guide microglia towards their targets. Now, a few years ago, people have shown that in the case of neuronal injuries, microglia are guided by ATP signaling, and they are able to pick up ATP thanks to purinergic receptors, in particular the P2Y12 receptors present on their plasma membranes. ATP, however, is a fast diffusible and rather unstable molecule in the extracellular space, so it's unlikely to provide long-lasting positional information by simply diffusing out of a point source, which in this case is represented by injured neurons. Therefore, people have also proposed an alternative model by which somehow cells along the path of migration do secrete ATP. In order to study these issues, we used the zebrafish embryonic brain for several reasons. First of all, this is optically transparent, it's very small and easily accessible. And most importantly, the microglia network is only formed by 25-30 cells that constitute a small group that is very easy to study.
To investigate the response of microglia to injury, we have used a pulsed UV laser coupled to a confocal microscope. Using this laser, we are able to induce robust and reproducible damage within the living brain. We found that killing of around 200 neurons at the midline attracts microglia, and the migration of the microglia is like in the mouse mediated via ATP that is sensed by the P2Y12 receptor expressed on the microglia. We got an entry point into the molecular mechanisms underlying the response of the microglia when having a look at calcium signaling in the brain. To do so, we generated a transgenic line that allows us to live image calcium levels within the living brain. And we saw that upon injury, there's a graded calcium wave that travels across the brain, and the response of the microglia is highly correlated to the calcium wave. And killing fewer neurons leads to a smaller calcium wave, and only the microglia in the direct proximity of the calcium wave respond to the injury.
To test if these graded calcium waves are necessary and sufficient to drive microglia, we have built a UV cutting laser and two mechanical arms that allow injection and uncaging under the microscope while imaging. In this way, it has been possible to uncage a calcium activator in a single neuron. In the absence of injury, microglia branch towards neurons with high calcium levels, but they don't if we knock down the P2Y12 receptor present on these cells. A big question, of course, is what promotes these calcium waves. We found that this is not ATP but glutamate acting via the NMDA receptors. In particular, we can uncage NMDA in the living brain, and this promotes calcium spreading and attracts microglia. Again, we abolish the microglia response if we hydrolyze ATP or knock down the P2Y12 receptor. So we show that glutamate diffusing out of the damaged neurons activates NMDA receptors along the path and promotes graded calcium levels in the surrounding tissue. This in turn promotes the release of ATP that binds to the P2Y12 receptor on microglia and ultimately guides these cells to the injury.
This is most likely not the only guidance system. So as, for example, in the P2Y12 knockout mouse, the response is delayed, but after several hours, the microglia reach the site of injury, suggesting that there is a backup mechanism that comes into play later on.
It's also going to be interesting to see how this molecular mechanism scales up and down with the damage size. In particularly, if only a couple of neurons die, only some microglia respond, so the signal must be restricted somehow in the brain.
Yeah. Also, is this a universal language because microglia are able to recognize different kind of neurons, and it's going to be interesting in the future to see if these cells communicate their status to microglia using distinct set of molecules, for example.